Welcome to 26-3, where we'll talk about how we actually make urine. So on the right-hand side, I have a picture here that I strongly recommend you recreate or add this detail to your previous picture where we just drew out the renal tubule and we identified corpuscle versus tubule and all the separate parts. But totally up to you on how you want to do it. Here I have the glomerulus in the renal corpuscle surrounded by simple squamous epithelium. Then I have the proximal convoluted tubule with cuboidal epithelium that has microvilli. Then the descending limb and ascending limb, with the descending limb having simple squamous, the ascending limb having a mix between simple squamous and cuboidal. And the distal convoluted tubule having cuboidal epithelium but with some microvilli. And the collecting duct having our two different specialized cuboidal cells. And then I have a key on the left side. So while you're drawing that out, I'm going to talk about the slides before we add onto this picture. So metabolic waste. So we have three that we primarily care about that we have no other mechanism of really reusing in the body, and that's urea, creatinine, and uric acid. So urea is formed by the breakdown of amino acids. So this is breaking down the monomers of proteins. Creatinine is the breakdown of creatine phosphate, which is really important during muscle contraction. And if you, we always normally break down some amounts of creatinine, like there's a normal level in your blood at any given time. But if you have too much in your urine, then that could be a sign of problems with the skeletal system. So that's why you'll take the urine test in order to determine creatinine levels. Uric acid, this is the breakdown and recycling of nitrogenous bases from RNA. So this is something that's happening all the time in all of our cells, and we don't really have a reason to keep uric acid, so we dispose of it in urine. But all three don't like being by themselves. They have to be dissolved in water so that way they're not toxic to the surrounding epithelium as they leave. So we have three pieces that we need to think of whenever we're talking about urine formation: filtration, reabsorption, and secretion. So filtration is primarily happening at the glomerulus. This is what the glomerulus will allow into the proximal convoluted tubule. That'll be filtration. We'll talk more about how the glomerulus is controlled in the next unit. Then we have reabsorption, and reabsorption means that anything that is in the renal tubule that we can reuse will be reabsorbed into the peritubular capillary network. So that's what we mean by reabsorption. And in the picture here on the right, I have that as a teal color. And then we talk about secretion, and secretion is in the opposite direction. So what do we not need from the peritubular capillary that we can dump into the tube? Or what's the difference in concentration gradients, and maybe it's just passively coming into the tube anyway. So reabsorption and secretion, don't get those two confused. And I have secretion in a salmon color over here on the right. So this is a good picture to kind of start with for this unit, or for this subunit. It gives you a good shortened version of what's really going on in each part of the nephron. The proximal convoluted tubule is going to do about 90% of the absorption or the reabsorption of the filtrate. The nephron loop is going to be primarily concerned with concentrating that filtrate. The distal convoluted tubule is primarily concerned with fine-tuning that filtrate. And then the collecting duct is primarily concerned with reabsorbing water. And then at the end, we finally make urine. This picture on the left is really nice because it gives you the arrows and the directionality of where things are coming and going. So this is a good starting point, or if at any point you get confused on the right-hand side, once we finish that picture, you can come back to this image and maybe that'll help reorient you. For now, I'm going to move over the lecture slides and just focus on our diagram here. Let's go ahead and start by identifying what is actually in filtrate. So filtrate is coming in from the glomerulus. Let's go ahead and identify what's in filtrate. So we'll start with urea, which we've talked about, creatinine, which we talked about, uric acid, which we talked about. Then we're going to have a lot of water, which we'll probably want to reabsorb. We'll have glucose, which we definitely want to reabsorb. We'll have potassium, which will somewhat reabsorb. We'll have sodium, which will definitely reabsorb. We have chlorine, which will exchange for some ions, but will reabsorb primarily. We have calcium, which will reabsorb. We have magnesium, which we don't really talk about, but I'm just adding that in here. We have, I think this is sulfate, sulfide, I'm not really sure, but this is also existing in the filtrate. This is just kind of passively floating back and forth. We have bicarbonate, which we'll definitely talk about, how that gets reabsorbed. We have amino acids, which are really small, so they can pass through the glomerulus, but we really shouldn't have a lot of these, otherwise that would be a bad sign. But remember, amino acids are the monomers of proteins. Speaking of proteins, we have very small, very small proteins that can also pass through the glomerulus, and we don't have any way of really reabsorbing these once they get through the glomerulus. So again, we don't want this, but there might be a little bit of some protein spillover. Then we have ammonia, excuse me, not pneumonia. And this is what primarily makes urine smell like urine. So we'll release that. Then we have what we've already learned from the blood chapter, urobilinogen. This is what's going to give us that yellow color of the urine. Then we have bilirubin if we didn't use it anywhere else, or we have an excessive breakdown of red blood cells that can leak into the urine as well. We'll have some neurotransmitters, which we won't really talk about, but again, this is just an excess. Some vitamins if they didn't get reabsorbed at any point or used, which is what the vitamin companies want you to buy. And then last but not least are drugs. So this is why you'll take urine tests, is because drugs have no way of being really reabsorbed at any point within the nephron. So once they pass through the nephron, they're staying in the urine. They're not going to get reabsorbed at any point. Okay, so that's filtrate. That's what's left the glomerulus. Now let's talk about what's happening at the proximal convoluted tubule. I'm going to start with what gets secreted into the tubular space. And the first thing I want to start with is hydrogen protons. So that's going to acidify the urine. Then we're going to primarily reabsorb everything else. So like I said, glucose is going to get reabsorbed. Sodium is going to get reabsorbed. Water is going to get reabsorbed. And then we're going to have CO2 also get reabsorbed. And remember, CO2 doesn't like hanging out just like as CO2. It likes to be converted into H2CO3 by carbonic anhydrase. And then again, it can be converted into hydrogen protons and bicarbonate, which in this case it gets turned, so I'm just going to put carbonic anhydrase here. In this case, it'll get converted into bicarb. But bicarb doesn't like going straight into the bloodstream by itself. It'll have to be co-transported with more sodium ions, so we'll have both of those coming together into the bloodstream. And I'm going to draw that circle there as in co-transport of those two. All right, that's it for the proximal convoluted tubule. Now we'll move down into the descending limb. I have a lot of simple squamous epithelium here. I don't have any cuboidal epithelium, which means I'm probably not going to be secreting anything in here. Also, simple squamous epithelium means that there's a lot of easy passive diffusion that can happen. So here we're only really concerned with water. And water, we had a lot get reabsorbed in the PCT, but we really want to concentrate the urine and hold on to water as much as possible. So we're going to get rid of water from the tubular space. So H2O, 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 and H2O. So all along the descending limb of the loop of, or the descending limb of the nephron loop. So by the time we get to the bottom of the nephron loop, we don't have a lot of water, we have some remaining solutes, which means we have a relatively high solute concentration. So our filtrate at this point is a really high solute concentration. And that's what the brackets mean. This is going to be a really important concept whenever we introduce countercurrent multiplication, which is toward the end of this chapter. So for right now, you need to understand that how we got to this high solute concentration is because we got rid of a lot of water in the descending limb, and that's part one of that story. We'll continue that story a little bit later. Okay, so now we go through the ascending limb. I still have some simple squamous epithelium, but then I convert into cuboidal epithelium. And here, we're going to be reabsorbing substances, and those two ions are going to be sodium and chloride. So sodium ions, chloride ions. And we do a relatively good job of this. We don't get rid of all of it, but we definitely want to hold on to it as much as possible. So we start absorbing solutes. And this makes sense because at the bottom of the nephron loop, we have primarily solutes over water. So by the time we get to the ascending limb, which by the way is not permeable to water, so we can't move water at all, however you want to draw that in there. We can only move solutes, and so this is a perfect way of reabsorbing those last bit of solutes that we really want to hold on to, like sodium and chloride. Okay, now we get up to the DCT. Let's start with the easiest one first. This is where we're actually going to absorb calcium. So I'm going to draw calcium 2 plus. Oh, I'm sorry, I'm going to draw it this way. It goes into the bloodstream, calcium. All right, that's the simplest one. Calcium gets reabsorbed at the DCT, and this is primarily coordinated by parathyroid hormone. So if you remember, parathyroid hormone is something we learned in the endocrine system chapter. It's released by the parathyroid glands, and this is promoting calcium reabsorption. Okay, next, let's talk about carbon dioxide again. So whatever leftover carbon dioxide, oh, and I'm sorry, I still meant to draw it. I drew it like that again. I meant to put calcium here. Sorry about that. Okay, so now let's talk about carbon dioxide again. So CO2, whatever remaining, again, we want to absorb it, but we don't, it doesn't usually stay in that form. So it gets converted to H2CO3, and as we know, H2CO3 gets changed to bicarbonate and hydrogen protons. And so bicarb will stay in the bloodstream to help buffer, but the hydrogen protons aren't going to go along with it. They are going to get secreted into the tubular fluid and create a more acidic environment. To exchange these hydrogen protons, we're also going to have an amount of sodium here, and sodium is going to want to go, or in order to exchange the hydrogen protons, we have to have a protein exchanger. So I'm going to draw one of those protein exchangers here. So we're going to be moving sodium in exchange for those hydrogen protons. Whenever we get the bicarbonate ion into the bloodstream, that doesn't like going into the bloodstream by itself. We have to exchange that also with chloride ions. And then another exchanger that we have, I'll draw down here, but we're going to exchange sodium for potassium. So we're going to put sodium back into the bloodstream, and we're going to exchange that for potassium. Okay, why do we care? So we like holding on to sodium, that's obvious, but we don't want too much potassium in the tubular fluid at any given time. And we still need some sodium protons, or we still need some sodium cations in order to actually exchange for hydrogen protons also. So in order to regulate this sodium-potassium exchanger, we need to use the hormone aldosterone. And aldosterone will promote this exchange. All right, so let's think about this for a second. So if aldosterone is turned on, and let me remind you where aldosterone is coming from. So aldosterone was stimulated by the RAS system. RAS system got turned on because we had a decrease in blood pressure. Kidneys released renin. We went through the RAS system. Then we got to the adrenal cortex. The adrenal cortex made aldosterone. And now the aldosterone is going to work on the distal convoluted tubule to promote sodium reabsorption. And we're going to this increases blood pressure because with increased sodium reabsorption comes water, which means we increase blood volume and increase venous return, and thus increase cardiac output, which increases blood pressure. Okay, so let's say aldosterone is cranked on because we need to increase our blood pressure for whatever reason. Then we're going to lose a lot of potassium ions. This could be potentially really dangerous because then we might end up in a, let's draw it in this color, so low potassium. We might end up with a hypokalemia or hypokalemic state. why are we hypo and not hyper? Well, because all of those potassium ions that were in the bloodstream are now going into the urine, and we've lost them. We can't reabsorb them at any point. So this could be potentially dangerous if aldosterone was left on for a long period of time. This could also be dangerous because if we have a large sodium concentration in the distal convoluted tubule, that will also increase blood pressure. But that exchanger will also promote a lot of hydrogen protons in the distal convoluted tubule, which will increase the acidity of the urine, and then that could be potentially dangerous just for the epithelial linings of the urinary tract as the urine actually exits. So we need to care about what aldosterone is doing down here. Okay, so we need to mention one other co-transporter, and that again is going to involve bicarbonate. So let me remind you that in the cells, this carbonic anhydrase is constantly working, and so we're going to move bicarbonate again into the bloodstream to help buffer. But as one of the byproducts of breaking down amino acids, one of the byproducts of that is also ammonia. And so ammonia is going to get pumped into the tubular fluid. And like I said, ammonia is the one that actually gives that urine-like smell to it. So we can write here that this is amino acid breakdown, AA breakdown. And this is a byproduct of that. But ammonia doesn't like going into the tube by itself. It has to be co-transported with chlorine as well. So again, we're moving more chlorine into the tubular fluid. Okay, so that's all that we care about for the distal convoluted tubule. Now let's move on to the collecting duct. So first things first, easiest piece, is let's talk about acid-base balance. So at this point, we have a pretty acidic environment, assuming that everything's turned on, but sometimes we might not have a really acidic environment. Regardless, we need to have a method to control this, so I'm going to just arbitrarily pick one of these cuboidal epithelial cells, and we might have an exchange of hydrogen protons or bicarbonate ions in order to maintain acid-base regulation. So that can happen here, not really in a large amount, but it can happen down here. The other thing that can happen down here is, again, we'll have another sodium-potassium exchanger. So potassium going in. Potassium going in and being exchanged for sodium. And just like with our other sodium-potassium exchanger, aldosterone is also going to control this as well. So this is an important note. Aldosterone doesn't only affect the distal convoluted tubule, but also affects the collecting duct. So it works on two different parts of the nephron. So aldosterone will continue to promote that exchange in the in the collecting duct. Okay, all right. Now, last but not least, in the collecting duct, we have to mention how we're going to reabsorb water. So let's just take one of these cells and kind of zoom in and take a look at what's actually going on right in here. So let's zoom in. And I'll draw a box. And here we'll have our basolateral side. And here we'll have our apical or luminal side. Okay. And another blood pressure hormone that we've learned about is ADH, antidiuretic hormone. And remember, ADH is released by the posterior pituitary in order to promote water reabsorption, which thus increases blood pressure. So here's how we're actually going to utilize that hormone. So ADH is floating around in the blood, travels down. It'll look for these cells in the collecting duct, and it'll come and talk to the basolateral side of those cells. Waiting for ADH on the basolateral side are going to be these water channels that are very specialized water channels, and these water channels are called aquaporins, which is a beautiful name. Aqua referring to water, porins because they literally turn into pores. And ADH will tell those aquaporins once they're activated. It'll tell those aquaporins, hey, you're needed. We need to get more water into the bloodstream. So it'll tell those aquaporins to move to the apical side. And once they move to the apical side, they'll open. And then they'll wait for water to float by. And when water floats by, it'll get brought back into the bloodstream, which is pretty cool. Let's go ahead and draw that here in the collecting duct. So we've got water floating around. We've got our aquaporins that were originally closed, but ADH came and told them to come to the apical side. And when they got to the apical side, they opened. And then whenever water passes by them, it gets reabsorbed and gets shot back into the bloodstream. Increases venous return, thus increases blood pressure. So that's how ADH is working on the nephron to promote increase in blood pressure. You'll want to make sure to update your hormone tables appropriately for both aldosterone and ADH and parathyroid hormone. So now let's talk about what we have finally made. So we've made urine, but what is actually in this urine? So we've made urine. That's great. Oop. So let's see here. Urine equals. So we've gotten rid of a lot of water, but we still have a little bit of water because, like we said at the beginning, creatinine, uric acid, and urea don't want to. They don't want to be. They want to be dissolved in water, so we're going to have that here as well. Creatinine, urea, uric acid. Awesome. We have ammonia, which is what makes that urine smell. Then we have a lot of potassium ions. We have some hydrogen protons. We have a little bit of calcium because we might not have done such a great job of reabsorbing it at the DCT. Maybe our hormones weren't in check. We've got some neurotransmitters because we didn't talk about how we got rid of those. We've got drugs because we can't get rid of those, both recreational and medicinal. We've got urobilinogen to make it yellow. We've got bilirubin. Hopefully we don't have high quantities of these, but if you have a lot of red blood cell breakdown, you might have some extra bilirubin in the urine. And that's it. That's it. We got all of them. All right. So now let's talk about where that urine goes once it's produced. So urine then ends up meeting at the papillary duct, goodness, which dumps into the renal papilla. Papilla, goodness. Then the renal papilla will dump into the ends of the renal pyramid. The renal pyramids will meet at the minor calyx. Minor calyxes meet up with the major calyx. The major calyxes meet up at the renal pelvis. The renal pelvis turns into the ureter. Ureter travels to store the urine in the urinary bladder. Then we're like, oh my gosh, I really have to pee, and so we release it through the urethra, hopefully voluntarily. Okay, all right, so what do I really need to know from this? Everything. So this is a really important concept, is really understanding how urine is formed, what is urine, and why what things are being reabsorbed and secreted into the tubular fluid. What does the glomerulus let into the PCT as filtrate? Why do we have to have the peritubular capillary wrapping around? So some things are moving from a high to a low concentration, but some things are getting reabsorbed just so that we can use them again. We'll talk again in a later section about why the nephron loop is really important and the organization of the nephron loop. But for now, you need to really understand what things are moving in and out before continuing on. So in the next section, we'll talk about how the glomerulus actually is how the glomerulus coordinates how much blood it receives and how much filtrated it can actually produce.